Hello, and thank you for joining me at this virtual presentation, and thank you for the organizers at Twist for arranging this great opportunity to speak to you. Today I'm going to talk to you about SARS coronavirus 2 and variants. And if you're like me, you've probably gone through a period of very good optimism, thanks to largely vaccination and how we've been able to start to put together probably a more closer type of lifestyle to what it was before the pandemic. But clearly, there have been change. The optimism has been tempered by the fact that new variants have emerged, most notably the Delta variant, that have perhaps made us question whether the coronavirus pandemic will continue or whether we will go back to normal sooner. And if you pay any attention to the news, it seems like almost every week there's a new headline about a new variant. For example, there's a Lambda variant, and then there was a Delta Plus, and then there's a highly increased and mutated variant out of South Africa. There's the new Mu variant, and there's even some guy talking about COVID-22. So when you see these headlines, it's very difficult for us to interpret what this means and whether we should remain optimistic or will we be headed back to the worst parts of the pandemic. And today I'm going to talk to you about how we've developed a tool that hopefully allows you to see these headlines and see the emergence of these new variants and perhaps find a way to properly respond. So one thing is for certain, the continual evolution and emergence of SARS-CoV-2 variants is almost for certain guaranteed. As this phylogenetic heat map shows you that while we started in December of 2019, that by this summer, there are all these new lineages of COVID variants that have emerged and spread globally. Now, when we talk about variants, they're really defined by mutations that are made. Amino acid changes to the various proteins in the spike protein in particular play a major and defining role. And here is pictured the spike protein and some signature mutations of some of the variants that have arisen. Now, when it comes to the spike protein, it is really the receptor-binding domain pictured here, and its interaction with the ACE2 host receptor. So the RBD, as its name implies, directly engages with the host receptor and plays a critical role in the actual entry of the virus into host cells and then its further proliferation. Consequentially, it turns out that blocking the interaction of the RBD with ACE2 with antibodies is one of the best ways in which our host immune response is able to neutralize infection. Accordingly, several therapeutic antibodies, as pictured here from Regeneron and Eli Lilly, have been developed as therapeutic antibody drugs, and they work by directly engaging regions of the RBD that block its ability to engage the host ACE2 receptor. Now, enough research has been done over the last year and a half to uncover that this RBD, now pictured in light gray, actually has several key epitopes. And many of the antibodies that the human population develops are basically categorized into what are referred to as classes or classification system that is based on 1, 2, 3, and potentially 4 sites. Now, there is, of course, some nuance here, and there may be different types of antibodies among these classes, but this is a nice way to sort of resolve how the immune system and how antibodies engage with ACE2. And accordingly, as we've tracked mutations to the RBD in new variants, it's become clear that certain signature key mutations that are found across different variants, like, for example, the alpha variant, formerly known as B117, B1351, which is the beta variant, and P1, which is the gamma variant, they all undergo a mutation in position 501 from an N to a Y. That is actually potentially driving increased binding to ACE2. It also, the beta and gamma variants have mutations in position 417 of the RBD, which apparently drives some escape from class 1 antibodies. And another very common mutation found both in the beta and gamma variants is E484K, which seems to drive escape from some class 1 and 2 antibodies. So the key to take away here is that mutations to the RBD play an important role in potentially causing escape from neutralizing antibodies. So something to consider is that as more and more mutations occur and new variants emerge, eventually there is the chance to basically resist vaccination, vaccine-induced immunity, and the antibodies that are developed. So if you look at this graph here, there was actually a landmark moment last November 2020 when the first therapeutic antibody from Eli Lilly called bamlanivab received FDA approval. However, by April 2021, just a few months later, the FDA had to revoke the approval for this drug because of the emergence of variants that had resisted this antibody almost altogether. And if you look at this graph on the far right here, what you can see are there are several of these new variants. And when we started with the original Wuhan version of the virus, this is data showing the serum immunity in an in vitro assay from patients who had received the Pfizer BioNTech vaccine. And so 1 week after the second dose, about 96% of the population of this cohort showed extremely strong neutralization. However, new variants like the mu variant and even the beta variant, this number drops dramatically. And so this is one of the keys to remember is that eventually current antibodies will lose efficacy to some of these novel variants. And that, of course, is what risks the trajectory of this pandemic and its prolongation. So some amazing research has been conducted in the last year, and much of it has been led by Jesse Bloom, who's at the Fred Hutchinson Cancer Center in Seattle. And what Jesse Bloom and his group developed is a yeast display platform where they express the RBD on the surface of yeast, and they generate single position mutagenesis libraries. This is a method called deep mutational scanning. So they basically make single mutations across the entire RBD domain, which is about 200 amino acids, and they screen it phenotypically by flow cytometry for RBD mutants that bind or escape to antibodies, as well as RBD mutations that bind or escape to the ACE2 receptor. And they generate really interesting and rich data sets that tell you about how particular mutations impact potential escape to antibodies, as well as escape maps that are pictured here. And obviously, Jesse Bloom and his group have been prolific at publishing this work, which shows you, exemplifies essentially the impact this method has had on the field. Now, one thing though to keep in mind though, however, is that the new variants that are emerging for SARS-CoV-2 do not have single mutations. In fact, many of them have multiple or combinatorial mutations in the RBD domain. Here is the new variant called C12, which just was reported in the summer to emerge out of South Africa, and it shows of up to 4 mutations in the RBD alone. So as this occurs, it means that there's really the technical challenge of how do we understand and predict the impact of multiple or combinatorial mutations in the RBD on both ACE2 binding, but also antibody escape. So to do this, my group developed a method that we call deep mutational learning, or DML. And it allows us to, what we believe is perform predictive profiling. And so it starts by looking at the RBD. So remember, the RBD is about 200 amino acids long, position 333 of the spike to 527. And if you look closer at the actual critical residues, there are certain sites of the RBD, sometimes referred to as the receptor-binding motif, which show a lot of genetic variation, which is what these colors represent. And they also show direct engagement with the ACE2 binding sites. And many of the variants, like the beta and gamma variants, have mutations in these particular sites. So what we did is we essentially created a yeast display library that introduced mutations into these critical sites of the RBD, but combinatorial mutations, not single point mutations. And then we screen them, these synthetic libraries of mutations, for binding to ACE2, to class 1, class 2, and class 3 antibodies. We chose essentially the 4 clinically approved antibodies from Eli Lilly and Regeneron. So we perform yeast display and flow cytometry sorting of the positive and negative fractions. We first sort for all ACE2 binding variants, and then the ACE2 positive fraction gets sorted for antibody binding or escape. And from that information, we perform, from those selections, we perform deep sequencing. And then from the deep sequencing, we train machine learning models that are used to try to predict the escape or binding of any given variant that emerges. And then we perform different types of analyses that try to look at the long-term trajectory of mutations on binding or escape. And I'll walk you through this now in more detail. So the first thing we had to do was design combinatorial mutagenesis libraries. And here is that region 484 to 505 that is pictured. And what these amino acid distributions represent is the design of the library. And we based this design actually on DMS data that Jesse Bloom had published last year. And it allows us to create a combinatorial library while also keeping some positions highly conserved, because the DMS data suggests that mutations in these regions are essential for ACE2 binding. So once we've created this combinatorial design, we then generated the library, and the theoretical diversity is around 6 times 10 to the 12, which is far greater than what you can experimentally screen in yeast, which is typically limited by about 10 to the 9. So once we created that, we also had to generate what we call a tiling mutagenesis library. And these libraries essentially have a small number of mutations. Basically, 3 degenerate codons are added and tiled across this region. And there's 220 oligonucleotides used. And this is to create a low distance mutagenesis library, which is actually essential for part of the machine learning workflow that I'll explain to you later. So, but all of these libraries were then experimentally generated and screened by yeast display. And what I want to show you in this flow cytometry to highlight is that this is binding to ACE2, is that each library has to have, that we sort it with a very clear population of the binding variants and the non-binding variants. We don't sort these intermediate variants, which actually have potentially some reduced binding or escape, because that would actually create a very noisy signal for the machine learning model. So having very clean populations is actually critical for the machine learning step. What these sequence logos now depict for you is essentially the variation in each of these libraries in the binding and non-binding fraction after performing next-gen sequencing. And what you can see is that by eye is that they actually look quite similar. The binding and non-binding fractions, while of course there's some changes to the distribution in some positions, one can certainly not look at this by eye and basically be able to identify a binder or a non-binder. And that's again shows you why the importance of machine learning will be necessary. So next we took all of the ACE2 binding fractions. So we actually take only the ACE2 positive binding fractions, and then we sort those for binding to or escape to the 4 clinically approved antibodies that are pictured here. Once again, we sort very clean populations. And then what you see here is pictured is a sequence logo of the 2C libraries. And again, you see a fairly similar looking pattern in both the binding and the non-binding fractions. Okay, so once all of that NGS data has been generated, the next step is actually to convert it into a machine learning model. And the first step of doing that is to encode the data. So we take amino acid sequences and we convert them through a process called one-hot encoding, which is just a matrix approach to essentially encode the data. And then from there, we take this encoded data and we have to essentially train a model with it. And we tried several different types of models, which I'll go through in the next slide. But what eventually we settle on is both two types of models, a random forest and a recurrent neural network. And we use this to essentially say what the model is going to do is it's going to take any given amino acid sequence of the RBD variant and make a prediction, a probability that it binds to ACE2 and a probability that it escapes or binds to a given antibody. And that is how essentially we can train this model on this experimental data to then predict eventually on in silico data. Okay, so when we set up this, we did first some benchmarking, and we tried a lot of common machine and deep learning models, simple models like k-nearest neighbors, linear SVM, logistic regression, etc. And you can see that they actually all perform pretty well for the accuracy score, especially the recurrent neural network, the RNN, and the SVM and the RF. The SVM is actually very slow to run, so we decided to discard it and only move forward with the random forest and the RNN models. And then a really important thing to highlight here is when we assess these models with the full training data, and we look across each distance from the wild type sequence, they perform extremely well. Even up to 10 mutations away from the wild type, we're still seeing really good performance from the RNN and RF models. Now, a contrast to that is if you actually tried to train machine learning models using DMS data, and remember DMS data is single point mutations, that they really can't do anything better than a random guess. They're essentially 50-50 for predicting binding to ACE2 that's pictured here. And the reason why is that DMS data just doesn't have enough mutations and sequence diversity to train anything that can predict anything farther away from, wild type. Okay. So once we established that we want to move forward with a random forest and an RNN, we had to evaluate the performance of this model. And one of the most common approaches to doing that is using something called a true positive and false positive rate by plotting true positive and false positive, what's called a ROC curve. And what you see here is that a better the model is, the closer the line is to having a very high true positive rate to false positive rate, the blue that's pictured here. The worse the model is, the closer you are to the diagonal, which is basically like a random guess. So when we assess the ACE2 positive model binding models, we could see that our models were extremely good. What's pictured here on the diagonal are when we shuffle the labels. That's basically the negative control, and those are random guesses as one would expect. So we actually end up an area under the curve greater than 97.97, which basically is the closer you are to 1, the better the model is. So it's a very accurate in models. The same is true for all of the models. So every single antibody we screen, we also train a model for those antibodies. And again, they also perform extremely well, well above 90% accuracy based on these true and false positive rates. So just to highlight why DML is very powerful, if we look at DMS data published by Jesse Bloom, you can see that this is the Regeneron antibody 10933. It doesn't show any particular escape mutation for E484K, looking at the escape map pictured here. There's also no escape mutations observed at position 490. Now, that's actually very good. In fact, the data from our data actually very nicely agrees with the data from Jesse Bloom's group in that both of our models predict for Regeneron 10933 no escape from E484K and F490. So here is basically the probability that the model gives that the binding is maintained. So the closer you are to 1, the more likely that the variant maintains binding to this antibody. So that is good. But the big advantage that our model does is that we can now look at the combination. And when you actually combine these two mutations, both our random forest and our RNN predicts escape of this antibody from these combinatorial mutations. And so this is just one anecdote, but it shows you now the potential that and the likelihood that single mutations are not predictive of combinatorial mutations, and what DMS cannot do with combinatorial mutations is possible with DML. Now, what we then did to highlight this even further is we took, so there are, hundreds of millions of possible sequences and variants that we can apply our models to. So we just decided to select rationally in silico generated sequence variants that are from this lineage of originally the wild type and then the alpha and beta and kappa variants. And as you're going down on this phylogenetic tree, you're adding more mutations to the RBD. And these labels show you that essentially this is what the model predicts. So the model predicts that there are a few variants that come out from beta that are 3 mutations away from wild type that maintain binding to ACE2, but then that start to escape some of the clinically approved antibodies. So so these are the model predictions. So then what we did is we actually expressed physically every single one of these 46 variants and we tested them. So when we tested these, it turns out that we were 92% accurate on our predictions. So essentially, the machine learning model was able to predict with high accuracy that these variants maintain binding to ACE2. There were a few that obviously failed, but otherwise, that's very good success rate. So if you consider that we are here at the beta variant, about 2 or 3 mutations away from wild type, that we did identify that through DML, that there are variants that are just 2 mutations away from the alpha variant that would actually give you escape from all 4 monoclonal antibodies. And so because of the 4 antibodies are a surrogate for potential vaccine-induced antibodies because they represent several classes, it tells you that if we ever were to see these variants, that would be of concern because they could have enhanced antibody escape and perhaps even vaccine resistance. Another very interesting thing is we could also take these sequences and we used AlphaFold to predict their structures. And what's rather interesting here is if you have the wild type here, you can see that we can create these mutations and that are predicted and confirmed experimentally to bind ACE2. And the AlphaFold predicts very unique and different structures from the wild type. What this indicates is, unfortunately, SARS-CoV-2 RBD has a lot of plasticity, meaning that it can acquire a lot of mutations and probably still maintain infection to host cells. So it has a lot of room to continually evolve and mutate. Now, one of the best applications of this technology is the fact that we need to predict how variants respond to given antibodies. And so Stanford has developed this really nice database of variants and basically if they escape from some of these antibodies. And that's what's pictured here. So they've retrospectively looked at experimental studies, data, and they've compiled this really nice database. But this database is small. It only has a small selected number of variants that have been tested. And it's mostly because it takes a lot of work to generate the experiments. So the real power of DML is that we can actually do all of this in silico. So what's pictured here is the alpha variant and every single, a huge panel of mutations that are distance 1 away from the alpha variant. And each one of the columns here, or rows, is depicting what our RNN or random forest model predicts for escape from one of these 4 clinically approved antibodies. So in most cases, the models do agree. So that's also a good way to benchmark. And basically, escape is when the value or the color is closer to blue. When it's red, it means the binding is maintained. So we can do this 1 mutation out. We can also do it 2 mutations out. We can go down and look at the beta variant. We can go with the gamma variant. And we can, of course, go many mutations out. I only picture 1 and 2 because that's probably the most likely mutations to occur, at least in the foreseeable future. So DML is really powerful because it allows you to do this prospective profiling on all of these mutational lineages all in silico without having to, of course, do all of the painstaking experiments. We can also look at these different variants and how many antibodies are escaping. So you can see as an example here, the gamma variant, by the time you get about 2 mutations out from beta or gamma, there's actually a significant fraction of possible variants that can actually escape all 4 of these clinically approved antibodies. And then the last thing that we can do is do something called what we call deep escape network. So this is a very complicated figure. So I'll try to walk you through it briefly. Basically, everything within this circle is a variant that has only 1 mutation from the alpha variant. and everything outside of the circle has 2 mutations. And then the different colors are indicating whether it escapes 1, 2, 3, or 4 of these clinically approved antibodies. And so you can see from the alpha variant, there's a small fraction of sequences that can escape all 4 antibodies pictured here in red. If we go to the beta and the gamma, you start to see a more significant fraction. In fact, the beta variant, there's 50%. It only takes 2 mutations for 50% of the variants then to escape to escape 3 of the 4 clinically approved antibodies. So again, this is quite a telling thing, and this allows us then to really understand how any given variant and how its impact as it continually evolves could have on binding to antibodies. So now if we come back to my first slide, where we talked about all of these headlines. So here's a headline from just about a couple of weeks ago with this new variant at the bottom here called B11523, which is claimed to potentially escape immune protection. So what DML allows us to do now is actually gives us a tool to immediately, rapidly determine the potential impact of this new variant. So we went in and we looked at this variant, and it has the 2 mutations described here in the RBD, E484K and S494P. DML predicts that this variant would actually escape the clinical antibody Li555. And then when we apply this entire predictive profiling, here you can see all of the Li55 variants that are 1 distance away escape, as that makes sense. Of course, it does as well for 2 mutations. But then you can start to see if this variant were ever to continually to mutate, that a significant fraction also escape other antibodies, like the other one from Eli Lilly and even one of the Regeneron antibodies. And eventually, if it were to acquire 2 mutations, there are some candidates here that escape potentially all 4 of these antibodies. So remember, this headline just appeared 2 weeks ago, and we could just easily go into the models and make these graphs and make these predictions. And of course, we can also look at the number, the fraction of antibodies, and then we can build this really nice escape network on B11523 and identify that, again, about 30% of 2 mutations would escape 3 antibodies, and about 2% of the 2 mutations would escape all 4 antibodies. Okay, to summarize, I think what hopefully I've shown you is that DML uses machine learning guided protein engineering to train very accurate models that allow us to profile the potential impact that SARS-CoV-2 variants have on antibody escape. They also allow us to identify this very highly diverse landscape of variants that maintain binding to ACE2. And it also allows us to perform prospective profiling of antibody escape. So essentially making a rapid prediction on emerging variants without necessarily having to go and do an experiment every time. We can already get a quick response. Long term, I think the outlook is that DML may allow us to guide us in the design of combination antibody therapies. We can essentially perform this process on new antibody drug candidates if they continue to be developed for COVID, and determine which combinations work best based on how much mutational coverage they have on prospective and future variants. And lastly, and perhaps most powerfully, is it may be possible to use DML on panels of antibodies, for example, like polyclonal antibodies or antibody from vaccinated serum samples, and really identify variants that already have potential for vaccine resistance. So as we move into the endemic phase of SARS coronavirus, it would be really important for us to understand this, because then it will, of course, maybe even allow us to understand how we should respond with whether it's booster injections or future vaccine versions of the vaccine. And here is just finally a nice little graph of of our deep escape network on the original Wuhan virus. So I want to thank this amazing team. So this whole project was a collaboration between my academic lab at ETH Zurich with some of the key notable people here who did all of the work, and a spin-off company, Deep CDR Biologics, in which I'm also an advisor and co-founder of. And Cedric Weber was the representative of Deep CDR that we work very closely with. I also want to thank the Botner Research Center for Child Health for providing funding for this project, and my whole entire research group who did an outstanding job in supporting this work. So I thank you for your attention, and I'm looking forward to the live question and answer session that is next.